Dr. Mary Ellen Sanders is an internationally recognized consultant in the area of probiotic microbiology, who for 30 years has helped numerous food and supplement companies develop and communicate about probiotic products. Co-founder and founding president of the International Scientific Association for Probiotics and Prebiotics, and chair of the USP Probiotics Expert Panel, she has also authored over 120 peer-reviewed scientific publications on efficacy substantiation, microbiology, and regulatory issues pertaining to probiotics. Dr. Sanders really is a probiotics champion. So Mary Ellen, thanks so much for joining us. It's great to see you.
Yeah, great to see you too, Stephen.
So let's start at the start, basically. So your Bachelor of Science and your Masters were in food science. So at what point did you discover the whole microbiome of probiotics and just think, this is for me?
Yeah. I really didn't get interested in probiotics until about 1990 when I went out to work on my own. So in graduate school, I did not work on probiotics. And I owe a huge debt of gratitude to Dr. Joe O'Donnell, who at that time was the executive director of the California Dairy Research Foundation. And he reached out to me when I started my own consulting business to author a general review paper on probiotics and health, what's known about probiotics and health. And it was a pretty big undertaking for me to do, but it really got me very familiar with the literature. Of course, what was available back then was way less than what is available now. And it was a unique article because no one had really done a comprehensive review of, from the point of view of what do probiotics tell us about, or what do we know about probiotic consumption and human health? And it really got me on the map. And there was not very many people in the United States doing what I was doing, offering consulting services. It got my name out there, and it really started things going.
So you did your PhD at North Carolina State.
Correct.
With Todd Kleinhammer, I think.
Correct.
So that was more on the dairy side of things, and
yep.
Okay.
Yeah, so my work really focused on looking at bacteriophage resistance phenotypes in lactic starter cultures, specifically in lactococci. And so Todd was very interested in developing, understanding the genetics of commercially important phenotypes. And bacteriophage resistance was one where there had been a certain level of instability with the phenotype. And so he had hypothesized that it might be linked to a plasmid genetic element. And so my research looked into that, and we found one. We found a plasmid-linked phage resistance trait in a lactococcus. And so it was a fun project.
So then you finished your PhD, and then you went out and did you go direct into forming a consultancy, or was there a period of like postdocs and?
No, I went to work for 6 years for Marshall Products. Basically, Miles Laboratories was who I worked for, and they did the research component for Marshall Products. They owned that business at the time. And Marshall Products back then sold dairy starter cultures. So I worked in the research group for 6 years with them. And then in 1989, Marshall Products was sold. So Miles sold them. They were going to move the business to Madison, Wisconsin. And so change was in the air. So my husband and I just decided at that time to move out to Colorado. So and that's when I started my consulting business.
Okay. So when you wrote this paper about probiotics initially, I mean, what was the research landscape like? I mean, was there much going on? There was no overall definition at that point. We're still 10, 11, 12 years ahead of the definition, right? So
well, yeah, ahead of the FAO definition. There certainly were published definitions, many of which contradicted each other. I think more or less people understood probiotics to be beneficial microbes that you would consume, orally consume, likely in food and dairy products. There was very little research out there, and what was out there, especially in terms of human trials, were not trials that were really conducted to the level that trials are expected to be conducted today. And it was very rare, for example, for a trial to even recognize or stipulate the strain designation of a particular product. If studies were done, they would pull products off the shelf and just talk about the product name. They wouldn't really identify or quantify the probiotics in it. And I would say, I mean, it was really at its infancy. Now, having said that, that's in a U.S. perspective. I mean, there was quite a bit more going on in Europe and in Japan at the time. But even then, the human clinical trials had a way to go. It was still largely a hypothesis.
So I do want to talk to you about the definitions.
Okay.
Because, so the FAO WHO definition that came out in 2001, 2002, how much of a turning point was that? Like, how important was it that we had this kind of globally recognized definition finally?
You know, that's a great question. I mean, to me, I think it was very important. These definitions are not trivial because they consolidate all the stakeholders around something that we can just kind of put a stake in the ground and say, okay, we're going to agree to this. And then you have a starting point and everything else can flow from there. And I don't think at the time I appreciated how important it was, but having that done under the auspices of FAO WHO, I think was really important. It brought a certain level of authoritative recognition to the definition, and I think put it on the map.
Mm-hmm. I mean, I know ISAP has kind of revisited it over the years, and you changed one word, so it shows that it's it stood up to the test of time.
Right. Yeah, the ISAP conclusion is that the definition basically needs to stand, but unfortunately, it's grammatically incorrect. So we had a long discussion with the editor of Nature Reviews Gastroenterology and Hepatology, and she's a tremendous editor. And we put the paper through, leaving the definition exactly as it was at the FAO paper. And she came back to us and she just said, it's not right. Like, it's you either need to put commas in there or and then it comes down to, well, we have this opportunity to publish a reconsideration of this definition. Yeah. We just decided we didn't think it was appropriate to miss that opportunity and to try to set it right. And so we changed a word and added commas.
So I did want to talk about ISAP because you co-founded it with Glenn Gibson back in 2000, 2001?
Yeah, it was actually incorporated in 2002 as a nonprofit organization, yeah.
So where did the idea come from that? I mean, how long had you been walking around with this idea in your head, or between you and Glenn?
Yeah, well, the idea started kind of as a reaction to what Jaro had proposed. So Jaro conceptualized IPA, and he viewed this as an organization and actually had approached me to be involved in the organization as an organization that was really going to be focused on industry and to kind of help promote, the quality in the industry. And they were very reasonable goals. But as he talked to me about it, I realized that what I was more interested in was an association that was focused on the science. And so I did not get involved in IPA, but it started me thinking, well, is there a possibility that this might be something that we should consider that really focuses, has a different focus than what Jaro had proposed. And it just so happened that Glenn and I were on an organizing committee for a symposium that was sponsored by Dannon, and we were sitting around the table along with Irene Lenoir-Wincoop, who was very instrumental in the early stages of thinking on this as well. And we just talked about it. And Glenn, of course, being the expert in prebiotics, and what I propose is, is there value to an organization that focuses on both probiotics and prebiotics? And both of them were very interested in it. And so what we did is we kind of agreed among the three of us, and then went to the symposium that was held in New York, and we convened the scientists that were present giving presentations at that symposium. We had a little room and had a little meeting, and I don't know, I have a photo from that meeting. There were probably about 25 people there, and they were all very enthusiastic, like, yeah, let's do it. And these are people on both sides of the ocean. And so we just kind of took it stepwise forward. And then, as I mentioned, we incorporated as a nonprofit in 2002. And then Gregor, being Gregor, was bold enough to just offer to host the first meeting. And that really got us started. If Gregor hadn't made that step forward, we might still be talking about it, but he was brave enough to go ahead and do it.
So I did want to go back to definitions again, because ISAP over the years has played a key role in not just revisiting the probiotics one, but the prebiotics one you revisited, I think, in 2017, and then very recently symbiotics. But I know that there is some frustration among some of your peers, and with how brands and journalists and many people are kind of misusing these definitions. So what can be done ultimately?
Yeah. Well, I think ISAP made a good first step in trying to consolidate these definitions, like pull together experts in the field and agree upon a definition. And I think once the scientific community can get on the same page about that, I feel that the industry and the regulators will follow. So we're not going to be able, as a scientific community, force anybody to comply with a particular definition. We're not going to be able to force anybody to quit publishing definitions that contradict or differ from the definitions that these consensus panels have come up with. But I just think in an organic way, it's going to work itself out as the field goes forward. And I do think that, as frustrating as it can be sometimes with regulatory bodies and their approach or reaction to the field, I think at the end of the day, they're driven by science. And that's why I think it's really important that these definitions come out of a scientific effort, because at the end of the day, I think the science will prevail. And the best we can do is to continue doing good science to move the field from less hypothesis and more rigorous documentation of the effects and the types of endpoints we're looking at, and it'll speak for itself. Yeah. Meanwhile, it's a difficult competitive environment because there's a lot of products that maybe aren't really up to snuff. But
yeah, so one of the things that I've heard ISAP discussing is around establishing adequate intakes for dietary microbes, which is a relatively recent conversation, but that would be significant if we were to arrive at that. So where are we with this? Are we still just talking about it, or is it moving?
Yeah, so we have had several board members that are very interested in this topic, and this has been kicked around, I'd say, for maybe 5 or 6 years, at least in my understanding, maybe even longer. But the idea has been published by a few different scientists over the past years, but it's basically just been offering a hypothesis that the consumption of live dietary microbes matters to health. So the next question is do they matter enough that there should be some type of dietary recommendation for them? And that's a huge step. So where we are is ISAP has picked this up, and we just have in press right now a paper with the Journal of Nutrition that's talking about our approach, and so hopefully that'll be out shortly. And we also presented a proposal to the ISAP Board of Directors to fund a study to interrogate the NHANES database to look for evidence of the consumption of foods containing live microbes and what that means in terms of the health status of the subjects in that study. So we consider that to be a fairly reasonable first step to say, is there any relationship? And what I feel is going to happen is we will either find something or we won't find anything. And then I think what comes from that, we will either find that there is data supporting the relationship or there's not. And if there's no data supporting the relationship, then what we have to decide is that because the data set isn't asking the right questions or isn't fully sufficient to help us develop this hypothesis. And if that's so, we need to map out, well, then what has to happen to be able to really answer this question? And so there's this whole scientific component of this to try to find out what level of evidence or what type of evidence exists between consumption of live microbes and health endpoints. Then there's following that is a whole component of this discussion that really gets into, well, what will it take to meet government requirements to actually get a statement into some kind of dietary recommendations? And there's a lot of interest in this. And so in addition to the ISAP group that has been kicking around for this, IPA reached out under Bruno Pott. And so Bruno and IPA and ISAP are coordinating or collaborating on this. I also sit on a committee, a gut microbiome committee with ILSI North America, and they're very interested in this. So we have a subcommittee that's dealing with this topic. And I asked Bruno to come onto that committee so that he could represent he could represent IPA as well as the LABIP and other potential Europe groups that might be interested. The idea is to open this up broadly, be as transparent as we can be, generate as much excitement and interest as possible. But pretty much everybody agrees that this will take 10 years or more.
Wow.
You're not going to see anything in the next couple of years on this. It's going to take a long time because chances are the studies we need haven't been done yet. We're going to have to find the money to do them. There's going to be a lot of steps. So this is not a short-term project. I'll be retired for a long time after we make real progress on this. But you have to start somewhere.
Right. And I'm very conscious that you're using the term dietary microbes. You're not saying probiotics. So this includes fermented foods and all of this.
Absolutely. And it includes non-fermented foods that carry live microbes. I was on a call yesterday with Bob Hutkins and Maria Marco, and we were talking about the methods we're going to use to ultimately assign values of microbes for the foods that are contained in NHANES. And, Maria is like, look, there's paper right here that says that washed spinach has 10 to the 7 CFU per gram, washed bagged spinach. And so it's like, well, okay, so we need to wrap ourselves around the fact that we consume microbes beyond just in a probiotic pill, a probiotic yogurt, or fermented foods. Yeah.
Wow.
Yeah, I know. It's not going to be easy. It is not going to be easy. And that's one of the things that I just love about ISAP is that we've got like these top level scientists that are donating their time to this, they just want to see it happen. And that's just how ISAP has always worked. Oh yeah, that's a good idea. Like, let's make that happen, and people are just all in.
Great. So I also wanted to talk about, so there's a lot of science going on, a lot of exciting science, let's say, that is capturing consumer interest, whether it's around gut-brain axis or potential weight management and all this kind of thing, which we can go on and talk about a little bit later. But I did want to talk about, some of the very important work that's kind of going on behind the scenes that for many people may not be sexy, let's say. If you're in it, it's probably sexy because that's what floats your boat. But I want to talk about the USP probiotics expert panel that you are chairing.
Yes.
And you've said previously, I'm going to use your words here. Consumers want to be assured of product quality, and that's what this panel is all about. So what's your view on the overall state of quality of products in the marketplace, and do we currently have the technology we need to truly assess it?
I don't have any quantitative information that can lead me to make a general statement about the overall quality of the probiotic products that are sitting on the shelves today. What I do know is there are a variety of papers that get published that do surveys in different geographical regions of different types of products. And they certainly suggest that there are problems with what's in the product matching with what's labeled on the product. And it's to what extent or what percent of the products are plagued with that or whatever, that's what I can't speak to. I don't know how huge the problem is, but I do think it's fair to say that the products on the marketplace are likely uneven in terms of at least how they're represented on their labels. And I think it's a critical issue. Labels should accurately reflect what's in the product, and they should provide consumers and healthcare providers sufficient information to be able to make reasonable judgments about whether or not they are interested in that product. And at a very basic level, it should have the genus, species, and strain designation of every strain that's in the product, and they should have colony forming units or some measure of potency through the end of shelf life. And I was involved with Dr. Dan Merenstein in some quick surveys that we did of product labels on the retail shelves in the Washington, D.C. area. And in a very general statement, over 50% of those products did not have that level of detail on the label, and that those are in the Washington, D.C. area. And so there's a real need for improvement. And that's where USP comes in. And USP can not only, or an organization like USP, they aren't the only ones that provide high level quality seals to assure third-party verification of product quality. There are other organizations that do it, but I just happen to work with USP. And where they can come in is they can audit the whole process and verify that quality is being built into the product. And then they can also verify that the identity and potency that's listed on the product label is accurate. And I think that would go a long way toward improving confidence that healthcare providers have about using these products, as well as consumers, in terms of being clear on what they're buying.
Yeah. I also wanted to ask you about CFUs very quickly, because again, I'm going to use your words, because we've got CFUs. The regulations, FDA still says just milligrams, right? I mean, there was that petition from IPA, which was accepted about CFUs on a label, but it's voluntary.
Right.
And then there's AFUs, which are now coming in, and you're starting to see on some products using that. And yeah, you'd said at a previous event, we do need a measure of potency that is not milligrams, but in the future, there's maybe AFUs, which is active fluorescent units, for those who don't know, or a technology we cannot predict at this time.
Right.
So there's two things. Is like, goes back to my question about, do we have the technology we need?
Mm-hmm.
I'm guessing the answer is no.
Oh, yeah.
And then also, this shows to me the kind of how far maybe some in the probiotics community are from where the regulatory agencies are. If you've still got milligrams, we all know regulators move really slowly. They only require milligrams, and yet the community is already talking about, well, maybe we need to go beyond CFUs to AFUs now.
Yeah.
What are your thoughts?
Well, yeah, and I think this is a really good point that you're bringing up. From my point of view, and this was the comment I had on the citizen petition that IPA filed, is that don't pigeonhole what technology has to be used. That if you go to the FDA, I think it's perfectly legitimate to tell them that listing milligrams on a probiotic as a measure of potency is nonsense. So they need to know that that's not okay. And I think the citizen petition served that purpose, but I do think that we need to be open-minded and realize that although CFU is the industry standard today, that may change down the road. And because regulators change so slowly, right at the upfront, the best thing, in my opinion, would be to just basically allow industry to come up with the appropriate potency measures that can be accepted broadly by scientific communities or whatever. So I think that's the situation we're in. CFU is the industry standard, and it's what's reported on essentially all clinical trials that have been published to date. And that is such a critical tie between the potency in the study that documents the health benefit and what's in the product. So it's going to be a tough move to get away from CFU. And so I think one thing that should be going on right now is anyone undertaking a clinical trial or a human trial, or in any host for that matter, should use both CFU and AFU as a potency measure, so that then if they want down the road to use AFUs to stipulate the potency of their product, they can prove that that's what the AFU was that was used in the study. Does that make sense, right? Yeah. So I think that has to happen. But I think I am not an expert in flow cytometry, but from what I understand, it's more precise and it's more accurate than CFU. And that's a huge improvement. And it's quicker. It's easier and cheaper to run, which is, again, very important. The difficulty is that there's not a direct correlation between AFU and CFU. There is, from what I understand, with fresh grown cells, but as soon as cells sit for a while and or go into stationary phase or whatever the different environmental conditions might be, those numbers deviate. And so I think we still have a challenge, as best I understand, to know how then to correlate AFUs to CFUs.
Is AFUs part of the conversation with the USP expert panel?
Oh, yeah. Yeah, we talk about it, yeah.
Okay. So now I wanted to flip back kind of just to last couple of minutes, just to talk about some of the science that's actually going on there around like the health endpoints. You and I have talked previously around, gut-brain axis and things like this. There's a lot of excitement. And consumers, I think, get it because, it's quite an easy thing for some people to understand, that when I'm stressed, I feel it in my stomach, and I think about it, and there's that, I get that link, that's
Okay.
easier for a consumer to get. But when you actually look at the science, a lot of it is still animal stage, right? There's not that much in humans. So it's almost like the marketers are getting ahead of
Right.
sciences. But as you look around, all of these new health endpoints that people are looking at, what is really exciting? Where do you kind of really sit up and say this is going to make a difference?
Yeah, well, I was laughing last night because I was just taking a few minutes to relax before going to bed, and I picked up my Equus magazine and I was reading it, and there on page 40 was an article, "Can probiotics help your horse?" And I thought that was pretty funny. It's like, wow, we've arrived. The high level performance horses are now being advised to take them. Um, it's a tough question for me to answer about where the excitement is, because you're right, there's so much going on. But I think, and especially after coming freshly off of this symbiotics panel, consensus panel effort that we put together, I think a really exciting area is going to be in added precision in using these substances. And there are a variety of ways to get added precision. and one of them would be to really design a true synergistic symbiotic where you have a prebiotic-like substance, whatever it happens to be, matched with a live microbe that together really bring about a specific effect, and it potentially at even low doses. Like you don't have, because you're feeding the microbe that you're looking at, maybe you don't need to provide 10 billion, maybe you can provide 200,000, who knows? You might be able to do very much lower doses of this, and you might be able to have, and because because a design like that allows you to pick the microbe that has the traits you need, as opposed to a prebiotic where you kind of have to, which I love the prebiotic approach, but you have to hope that the microbes that are going to do what you need them to do are present in your resident microflora. And so I think this added precision is really important. And I think it's important too, because from the point of view of understanding the role that our resident microbes play in efficacy. We know you can look at basically any probiotic trial and you see groups of people who respond and groups of people who don't respond. What is driving that? And we don't have a good sense of it. And it could be resident microbes, it could be diet, it could be other underlying, it could be underlying health conditions. There could be a variety of factors that might control that. But I think as the field moves forward, we're going to be able to get more precise. And I think that's going to feed in a little bit more to the drug model, the live biotherapeutic model, but not exclusively. I think there's plenty of opportunity for foods to explore those avenues as well. But I think that we're going to see developments in that area, and I think we're going to get better effects with less input down the road.
That's wonderful. Mary Ellen, thank you so much. It's been a pleasure talking to you today.
Yes, so I appreciate the opportunity to share these thoughts with you. I really do.
Right. Well, thanks for joining us, and take care of yourself.
All right. Take care.